Hi, this is the Acton-Boxborough Regional High School BioBuilders team, and our project this year is a bioluminescent Legionella detector. Legionella is a common waterborne bacteria that contaminates drinking water, causing a deadly type of pneumonia called Legionnaire's disease. To combat the spread of Legionnaire's disease, our solution is to engineer E. coli to detect Legionella in water and report using bioluminescence. Once Legionella is detected by an E. coli strain, a promoter will turn on the Lux operon, which will produce luciferase and luciferin, two substances that will react to produce a bioluminescent light. If a light is observed, then that means that Legionella is in the drinking water. As you can see, we were unable to do an experiment this year, but hope to do so in the future. Thank you.
Hello everyone, thanks for tuning in tonight. My name is Jeffrey Connors, joining you from the Andover team. And today I would like to explain our solution for coral bleaching. But before we get into that, I would like to introduce the rest of the team.
I'm Nadia Shah.
I'm Ava McCone.
And I'm Abea Javedi.
Home to billions of organisms, coral reefs are one of the most biodiverse places in the world. However, recently, due to the rise in ocean temperatures and fluctuating pH levels, coral bleaching, or the ejection of zooxanthellae from coral, has become more prominent. This in turn not only impacts the billion of organisms that live in reefs, but the many people who rely on them for a living.
For the reasons mentioned earlier, it is clear that it is not only in the best interest of the environment to maintain coral reefs, but also for the people that depend on them. As a result, we propose the following solution: to make zooxanthellae more tolerant to ocean acidification in warmer waters.
To do so, we plan to create a transgenic microalgae with our chassis as Symbiotium algae that will have a symbiotic relationship with the zooxanthellae. This process will be initiated with temperature-induced promoter.
When the temperature of the water rises for a period of 4 weeks or longer, this GMO will express more heat shock proteins. Heat shock proteins, or HSPs, are a group of proteins that are produced when cells are exposed to various types of stress. When exposed to thermal stress, such as an increase in temperature, HSPs have the potential to regulate the sensitivity of cells.
In our case, the GMO should specifically express HSP70 because it is well known to protect cells from thermal stress. When the transgenic algae produces the heat shock proteins, they will be contained inside the cell. Hopefully, our construct will provide an ethically and environmentally friendly solution to the pressing issue of coral bleaching.
Effect of UV light on antibiotic sensitivity. Our project was on antibiotic sensitivity, and specifically, we wanted to test the effects of ultraviolet light on antibiotic sensitivity. Antibiotics are antimicrobial substances that are effective against bacteria. Different kinds of antibiotics kill different kinds of bacteria, and the effects each antibiotic has on a specific type of bacteria can vary. To test which antibiotic is most effective against a specific type of bacteria, an antibiotic sensitivity test is conducted. In an antibiotic sensitivity test, we determine the susceptibility of a bacteria to an antibiotic. To conduct this test, we would spread out the bacteria we are testing on a petri dish using a cotton swab, and then place hole puncher discs that are coated with different antibiotics on different parts of the plates as well. The bacteria that are sensitive to a specific type of antibiotic would not grow near that disc and therefore would form a zone of inhibition, showing us that the bactericide is effective.
In our experiment, we followed the previous process and radiated one of them with UV light for 15 minutes while leaving the other one as it is as control. After 15 minutes, we put both discs in an incubator at room temperature, which is 25 degrees Celsius, and left it for 36 hours. Unfortunately, we did not see any bacterial growth in a dish that was irradiated with UV light, which tells us that the long exposure to UV light had killed all the bacteria.
Hello, we are the Baby Glen BioBuilders team. This year, we are researching remediating the impacts microplastics have on aquatic ecosystems.
Microplastics are tiny pieces of plastics that come from various sources. Many times they end up in aquatic ecosystems, posing health risks to many communities, as plastic is durable and easily accumulates.
The statistics behind microplastics are devastating. Every day, nearly 8 million pieces of plastic are thrown into water bodies. There are currently around 5.25 trillion macro and microplastic pieces in the ocean, and plastics make up to 60 to 90% of all marine debris studied. According to recent studies, a wide percentage of marine species have been found with traces of marine plastic pollution inside them as shown. In a single year, over 1 million seabirds and mammals are killed by marine plastic pollution.
This year, we have been researching how to remediate the impacts of this issue. We hope to see you at our presentation. Thank you.
Hello, this is Juan.
Hello, this is Alexander.
Our project is on a fire gel biofilm. The goal is to create a biofilm capable of releasing a fire-resistant gel that will aid firefighters in slowing down the movement of wildfires. This biofilm will be placed on trees located in populated areas and will protect life and property.
As seen in the diagram on the top right, there we have the design abstraction for a proposed cell. This cell will contain a heat detector which will trigger the cell to begin reproducing as well as begin producing and releasing the fire-resistant gel.
Before constructing the new cell, we have designed a preliminary experiment that will serve as the foundation for our cell design. Due to the large amounts of smoke in the environment our cell will be placed in, we think it is best to test the effects of smoke on cell growth. This experiment will be a good indicator as to whether or not the cells will continue to reproduce. The materials needed include several plates, bacteria, unscented incense sticks, and the lighter.
To begin, we will have 4 different treatments which we will test. A control which will get no exposure to smoke, then 3 other treatments with 10 seconds, 30 seconds, and 1 minute of smoke exposure, respectively. First, we will add the bacteria into the plates and allow them to grow. After days of growth, we will record the initial number of living colonies in each plate. Next, we will light the incense sticks and hold them close to the plates with the lids open for the set amount of time for each, as seen in the bottom right diagram. After a few days, we will remove the lids and count the new number of living colonies.
Unfortunately, because of school safety rules, we aren't able to replicate the environment of a forest fire like we would have liked to. However, we have tried our best to design our experiment using alternative methods such as this one.
We believe that with this preliminary experiment, we will gain a better understanding of how bacteria reacts with certain aspects of forest fires before we get to modifying it. In turn, this will help us get one step closer to our end goal of creating a fire gel biofilm to prevent the catastrophic damages fires may bring.
Thank you for listening.
Hello, we are the Blanton Jaguars presenting Bacteria Phosphorus. My name is Vu Fan. I'm here with my group members Martin and Zylene. Our objective for this project is to create a bacteria that could detect concentrations of nitrogen, phosphorus, and potassium, otherwise known as NPK, in soil with a focus on phosphorus. This bacteria could be used to assist farmers in providing the plant with adequate amounts of fertilizer for optimal growth. In our plan, we want to make the bacteria glow when there is low concentrations of phosphorus and stop glowing when there is a good amount of phosphorus in the soil.
Going into the second phase of BioBuilder, we learned very quickly we had an idea but not the qualifications or mindset required to continue with it. So we decided to come up with a different project as a way to gain laboratory skills and have more time to do better research. A project that would test how bacteria would react to phosphorus through the use of the BioBuilder kit we received. We decided to use the bacteria that came with it, Bacillus cereus, and the agar plate to design a project that would test how the bacteria reacts to different concentrations of phosphorus. We plan on dividing the plate into 4 parts, keeping one as a control and the other 3 as individual groups that would be exposed to different concentrations of phosphorus.
The method we have designed is to divide an agar plate into 4 sections and then go clockwise to determine the group number. We will then drip different concentrations in group 2 and 4 using sterilized pipette and then use cotton swabs to split the bacteria on the agar plate. The concentrations we have decided on using were the upper and lower limits of phosphorus detection found by previous studies who have also developed a bacteria that can detect phosphorus. Then we base the middle value on multiples of the lower limits of the phosphorus concentrations. Overall, with this project, we hope to gain more knowledge on what it takes to grow and handle a bacteria colony, so that we could plan to create this phosphorus detecting bacteria.
Title of our project is Bamboo Water Filtration System with Air Toxin Detection and Purification. The object of this project is to create a filter system that will notify you that there are harmful substances in the air and convert them into a neutral substance. The system will also purify water, serve as a carbon sink, and create ethanol for fuel. Techniques will be mutagenesis, transformation, PCR, microbial, culture techniques, and the CRISPR system. The CRISPR system will be used to insert a gene that will prevent the overgrowth of bamboo through turning on contact inhibition genes similar to those found in eukaryotic cells. This will prevent the budding of new shoots that will overcrowd the system.
Mutagenesis will be used in epsilon proteobacteria to insert the GFT gene that will generate fluorescence if the bacteria is actively metabolizing benzene, which is a toxin produced from gasoline production, into a nitrate, which will be detected in a fluorescence camera. This bacteria will be added to a kombucha-style fermenter that will produce ethanol that can be harvested for fuel. The limitations are cost, time, and ethical dilemmas.
Good afternoon. We are representing Masterman High School from the Philadelphia region. We decided to focus our research on asbestos as it is a hazard that directly affects us and the schools in our district. Asbestos is a carcinogen and can cause long-term health problems when inhaled. Our project aims to find a more efficient way to detect asbestos, specifically focusing on chrysotile, the most common form of asbestos.
Our design includes spraying DKSA protein bonded to the horseradish peroxidase on the possible asbestos surface. If the surface does contain asbestos, the DKSA protein with the enzyme will bind to the asbestos. After washing off the excess DKSA, we can spray in a combined mixture of ABTS buffer with ABTS substrate onto the surface. If we witness a color change from a very light green to a dark green, then we can confirm that there is asbestos present on the exterior.
Because of the recent pandemic, our team was unable to meet in person and conduct any variation of this experiment. So in other words, the full scope of issues or challenges that might arise from this experiment is not necessarily known at this moment.
And for a limitation within the project itself, because of asbestos's absorbent nature, the color green from the enzyme horseradish peroxidase might not necessarily be visible or easily detectable.
Because of how dangerous asbestos is, it's made impossible for students like us to gain access to it. What we recommend for teams in the future that decide to take on this project is to attempt to apply the concepts provided in this presentation to a similar but less harmful material, such as kaolinite clay. This clay is an important industrial mineral that shares a similar chemical composition with asbestos. In fact, the only difference between the chemical formulas of these two materials is the first element, making it a sufficient enough substitute to use.
Microplastics are fragments of any type of plastic less than 5 millimeters in length that come from a variety of sources, including from larger plastic debris that degrade into smaller segments. They can be harmful to our ocean and aquatic life. It is estimated that up to 13 million metric tons of plastic end up in the ocean each year. Fish, seabirds, sea turtles, and marine mammals can become entangled in or ingest plastic debris, causing suffocation, starvation, and drowning.
They can block the digestive tracts of marine life, diminishing their urge to eat and alter feeding behavior, all of which reduce growth and reproductive output. While plastics are estimated to take up to hundreds of years to fully decompose, some of them can break down much quicker into tinier secondary microplastics, which in turn end up in the seafood humans consume.
Our objective is to combat this microplastic pollution through the use of E. coli. A plasmid will be constructed using bioblocks, which are pre-assembled nucleotide sequences of the genes ISF6-4831 and ISF6-0224. That plasmid will then be inserted into E. coli, and successful transformation and insertion of our desired genes will be detected by luminescence.
A specific transcription factor for our system is a monomer produced from the breakdown of plastics. This was caused by a separate enzyme in our bacterial plasmid. The monomer, or transcription factor, binds to the bacterial DNA at the promoter upstream of the quorum-sensing gene of interest. This binding attracts the RNA polymerase to the site, thus initiating transcription of mRNA, which contains the quorum-sensing gene. mRNA is then translated into the quorum-sensing proteins. When quorum is reached, visible detection of light will report successful breakdown of polymer.
Though certainly advantageous in many ways, our project is not without limitations, which include access to necessary materials and equipment to carry out our experiment, limited vigor of response in E. coli to environmental stimuli, and the feasibility of inserting this gene into bacteria.
Thank you for listening, and please take a look at our poster to learn more about our solution.
How do you like your water? Whether your preference is bottled, sparkling, or even flavored, it is certain that we all like our water to some degree crystal clear with a pH of 7. At sites of abandoned metal mines, however, water is a bright orange color with dangerously high concentrations of sulfuric acid. This is due to the phenomenon of acid mine drainage, in which sulfide minerals, part of metal ores, are exposed to arid water, leading to the formation of sulfuric acid in these bodies of water. This drastically reduces the environmental pH, making the water inhabitable for aquatic life and harmful in contact with sources of water for nearby towns. Looking at all the reactions involved with acid mine drainage, our BioBuilders team recognized that certain reactions, catalyzed by acidophilic bacterial communities, increase the production of sulfuric acid. One of these reactions was the oxidation of iron from Fe2+ or ferrous ions to Fe3+ ferric ions, and it is facilitated by Acidithiobacillus ferrooxidans, an acidophile extensively researched in its contribution to acid mine drainage. Using databases like UniProt, Brenda, and the iGEM Registry of Parts, we identified the enzymes and DNA sequences involved in producing Fe3+, and one of these enzymes is known as iron resocyanate reductase. While our team was unable to conduct an experiment this year, if we could, we would try to remove the CYC2 gene that encodes for iron resocyanate reductase using the gene knockout method developed by Datsenko and Warner. Although more commonly used on E. coli, we believe that this gene inactivation method can help us develop Acidithiobacillus ferrooxidans bacteria that do not contribute to acid mine drainage.
"2020 was a year full of extreme weather-related problems around the world. We've had hot and dry conditions create record-setting wildfires across the world, unseasonably large Atlantic hurricanes wreaking havoc when they reached landfall, droughts that greatly harmed agricultural output in nations that already suffer from hunger problems, and several other issues across the globe. Of the many problems caused by today's climate crisis, melting glaciers and polar ice caps are one of the many long-standing predicaments caused by climate change. Left unchecked, these melting bodies of ice will add to rising sea levels, thus increasing coastal erosion and disrupting weather patterns. As a result, coastal communities and industries will suffer due to frequent flooding and more intense storms, along with wildlife that thrive in these colder climates. As a response to this global issue, the objective of this project was to create a solution for melting polar ice caps. To elaborate, we set out to find a way to decrease or completely negate the rate at which ice melts from the ice caps by using ice-nucleating bacteria to recreate the lost ice. In doing this, we'll be able to effectively reduce the impact of climate change and its effect on our seas.
Over the past several years, the number of dead zones in the ocean is growing exponentially, in no small part due to plastic pollution hurting the environment and its wildlife. Polyethylene terephthalate, also known as PET, is a widely used plastic material that's lightweight, strong, and often transparent. $34 billion plastic PET bottles produced and sold by the soft drink industry enter the ocean each year, despite the fact that the polymer is actually highly recyclable. Make use of some of the harmful waste. However, Most enzymes researched so far have proven to be moderately inefficient in hydrolytic capabilities. Last year, however, the French biotech company Carbios created an enzyme originally discovered in leaf compost that was capable of degrading plastic bottles for recycling in a matter of hours. In this project, we plan an experiment to combine this finding with another technique studied for PET degrading enzymes, which is the fusion of proteins to other proteins known as hydrophobins, assisting in hydrolysis. This will be facilitated through gene expression in bacteria cultures after transformation with an engineered plasmid.
As far as human technology is concerned, we still have not been able to fully cure cancer.
However, recent scientific and technological developments have led to the discovery of CAR T-cell treatment, which are now often used to treat blood cancers, such as leukemia.
CAR T cells work by using a form of immunotherapy which uses the patient's immune system's antibodies to attack and destroy tumors by targeting a specific tumor. This means that there is a lesser chance of the healthy cells getting killed or destroyed in the process.
Treatment starts from drawing blood from patients and separating the T cells, using virus vectors to deliver genetic material to produce receptors on their surface or chimeric antigen receptors. T cells are inserted. The T cells then recognize and attach to a specific tumor cell. The T cell then proliferates and becomes cytotoxic, which kills and destroys the tumor or cancer cells. 3 ways CAR T cells destroy our cells are through extensive stimulated cell proliferation, releasing of toxins which destroy the cells such as perforins and granzymes, and an increased secretion of factors which affect other cells, such as cytokines, interleukins, and other growth factors.
Our project aims to develop CAR T cells to penetrate the blood-brain barrier in order for it to work on brain tumors, as well as making it work on other immunology disorders.
For our ideas, a potential antigen we can target for our brain tumors are the SV40 antigen present in a variety of human neoplasms, such as mesothelioma, osteosarcoma, and brain tumors. This will be a potential antigen that can be targeted using CAR T cells.
Greetings, my name is Chris and Yuton Lee, and I study in a school called Sinarmas World Academy in Indonesia. So my topic for this project is called Plastic Fueled Microbial Fuel Cell, which is basically an alternative way and a more sustainable way to generate electricity while being able to reduce plastic waste. So the general idea is that I will be using a bacteria called Ideonella to break down the plastic and use another bacteria to use a substance that is broken down by Ideonella to as a energy source to generate electricity. Over the past few months, I have already made a working microbial fuel cell, but it was only using other bacteria, not Ideonella. And right now, I have been using Clustal Omega to compare 16 RNA sequence between different bacteria of known power density. As you can see in the picture in the bottom right, we can see that there's a list of bacteria with a list of the power density and their power density. So I'm planning to use this list and create a phylogenetic tree to compare these bacteria with Ideonella. And using this, I will be able to give out an estimate of the power density of Ideonella. Right now, because of COVID-19, I am facing a lot of limitations, and one of these limitations is that it is rather difficult to obtain the bacteria that I need, which is Ideonella as they don't sell it in Indonesia. And that will be it. Thank you.
The data I've come across in research papers and also collected from my own preliminary experiments support the hypothesis that Pleurotus ostreatus can be used to degrade single-use plastic bags, which are typically made of high-density or low-density polyethylene. As we know, single-use plastic and man-made plastic in general take a very, very long period of time to degrade, and as a pollutant to all environments, it is very damaging. A research paper titled Degradation of Oxo-Biodegradable Plastic by Pleurotus ostreatus showed in their results that it was highly probable that lacase contributed to the degradation of their plastic bags. This was investigated and tested through multiple techniques, much beyond physical degradation. As such, lacase is the enzyme being pursued, and since there are currently lab limitations, unfortunately due to the pandemic, I've continued my investigation by using the CASI database to study and analyze currently identified lacases in Pleurotus ostreatus. From here, I use distance trees, which are formed from blasting, to analyze the uniqueness and similarities between these lacases. If the data generates many similarities between the different types of lacases among Pleurotus ostreatus, this may mean that there is a characteristic lacase that contributes to this plastic degradation. Or, if there is a large variation and little similarity between the lacases, there is a very large chance that one of these many lacases have the ability to degrade plastic. In the future, these may support ideas of designs that may potentially produce more lacase for experimentation with plastic.
The objective of our project was to create a yeast that reproduces spider silk bandage when placed on a wound. The yeast Saccharomyces cerevisiae was an ideal chassis for many reasons. First, it could be easily transformed, allowing for deletion or addition of new genes for protein production. Secondly, it is better than bacteria at producing large proteins like the spider silk proteins in great quantities. Lastly, the Y55 strain was chosen because it has been identified as avirulent, unlike other strains that have been known to cause infection in humans. Components of two different promoters, AMB1 and AHPCP2D1, control the spider silk production.
The microenvironment of a wound is critical to the way it develops and heals. A lack of oxygen means the wound heals very slowly or not at all. So, a potential promoter is the yeast ANB1 promoter. Under aerobic conditions, the gene is suppressed and silk is not produced in the wound. Under anaerobic conditions, the gene is expressed so that silk is produced during the crucial time of low oxygenation. If the wound microenvironment were at sufficient oxygen levels, spider silk would not need to be produced since the wound would heal well enough on its own.
Reactive oxygen species, ROS, are radical derivatives of oxygen that are highly reactive and have added electrons. At a wound site, they function as secondary messenger signaling molecules that signal for hemostasis, white blood cell recruitment, and tissue repair. They have a crucial role in wound healing and therefore are prevalent at a wound site. This promoter, called AHPCP2D1, is regulated by a transcription factor called OxyR, which is activated by the presence of ROS, which is being used to pinpoint the location of the wound.
The AOX1 part of the promoter will ensure that the silk is excreted by the yeast so it doesn't build up inside. The CACG motif in the promoter enhances the expression of PYSP1. This motif has been found in the promoters for many species spider silk genes. Since each type of spider produces many types of silk, each different protein, we settled on AR-Genta's piriform silk to be created. While not the most well-known, this is the sticky silk spiders use to catch victims and securely attach their webs to low-energy surfaces, so this ensures the bandage will attach to skin. In addition, a little piriform silk possesses extreme strength, is biodegradable, supports self-assembly, and is like cement. It begins as liquid, but after a second it forms an immovable monoblock, perfect for a bandage.
For the silk to have antimicrobial and wound healing properties, we could insert the gene DEFB103B to code for the production of human beta-defensin 103B, or HBD3. The HBD3 induces secretion of the cytokine IL-17, normally found in human skin, but the beta-defensin would increase its secretion. When IL-17 is secreted, it induces AMPs, or antimicrobial peptides, that are already present in epithelial cells and increases their effectiveness. These AMPs inhibit bacteria and microgrowth while promoting wound healing in skin.
Hello, this is the Tyngsboro BioBuilder Club, and our project is AliveScent. Our goal for this year is to hatch and raise mosquitoes to test the effectiveness of limonene as a mosquito repellent. This is the next step to reach our original goal, to engineer E. coli to smell like lemons and act as a natural mosquito repellent. Our plasmid design has been changed from the last season to include two devices. Device A, as shown in the slide, is designed with a constitutive promoter
and a geranyl pyrophosphate synthase translational unit that will be assembled into a red fluorescent protein encoding vector. Design B is designed with a stationary phase promoter and a limonene synthase translational unit that will be assembled into a red fluorescent protein encoding vector. As of now, we are looking at plasmid PGL403 from the AdGene plasmid library for inspiration as we modify our existing plasmid because it contains both GPPS and LIMS, which are necessary for limonene production. Ultimately, we would like to test our genetic system using live mosquitoes. To that end, we have established a mosquito nursery for the breeding and testing of our system. To date, we have encountered difficulties in hatching due to egg viability and/or other environmental factors, but we are optimistic. After receiving the sensitivity disc kit, we thought it might be interesting to test the antimicrobial effects of limonene in a variety of other essential oils on Bacillus cereus growth inhibition. Interestingly, our results show that smaller concentrations of limonene are more effective at inhibiting bacterial growth compared to higher concentrations as depicted in our graph. Lavender was shown to be the most effective, prompting us to consider testing its effects on mosquitoes as well. We have plans to hatch a new batch of mosquito eggs and begin testing during the spring. Thank you.
Hi, this is Viggy from the Westboro High School BioBuilders team. Our mentor was Dr. Josh Atkinson, and our project is CalFarts, the sequel. So this for us is a continuation of our project from last year, in which we're combating climate change with synthetic biology, and more specifically, methane production from farmed cows, which is a pretty big contributor to greenhouse gas emissions. So early on into our process, however, we ran into some problems with methane, so we decided to shift our focus into sensing methanol, which is one part of that metabolic pathway. So essentially, we're trying to engineer E. coli to become a biosensor, which will sense methanol and then glow green when methanol is sensed. And we separated that into two separate parts, the sensing part and the glowing part, and we constructed a plasmid for each of those parts. In our sensing plasmid, we created a chimeric histidine kinase, which is a fusion of two histidine kinase sensors from Paracoccus nigricans and E. coli. We used the sensing domain FLHS from the nigricans and the kinase domain ENVZ from E. coli. And then in the plasmid that confers the glowing ability, we hooked up the Lux operon to the OMPC promoter. So at the end of the day, we have methanol being sensed by FLHS, creating a phosphorylation cascade signal transduction pathway, which turns on the promoter and then eventually the Lux gene. So at the end of the day, we essentially have E. coli, which senses methanol and then glows. We are still designing our future testing scheme, but right now we're thinking of having a negative control in which there is no pathway, there's just no bioluminescence. We're thinking of having a positive control in which there's always on bioluminescence using a constitutive promoter, and then our experimental group with our two plasmids. Thank you.
Hi, we're Weston High School, and this is our project, removing lipid stains on fabrics using oleate hydratase.
The purpose of our project was to develop an enzyme-based process that can degrade laundry stains more effectively and sustainably than current cleaning agents. One specific type of stain we decided to focus on are lipid-based stains because current detergents that degrade lipid-based stains are either not very effective or require non-polar solvents, which are either not very safe for all fabrics or are not environmentally friendly.
We set out to do this project so that clothes with stains could be cleaned and reused instead of getting thrown out. We also wanted to make removing stains an easier and less time-consuming task.
We plan to add a hydroxyl functional group to oleic acid in order to increase its solubility in water. Oleic acid is one of the most common natural fatty acids that is present in many foods. By adding a hydroxyl group using oleate hydratase, an enzyme, polarity is increased, and since we are using water as a solvent which is also polar, oleic acid can then be dissolved and rinsed away effectively.
We planned on using restriction enzymes to create two sticky ends on both the plasmid and the gene that codes for hydratase. Then we would combine these two fragments together to form the recombinant plasmid.
After we create the recombinant plasmid, we need to insert it into the bacteria through a process called transformation. To do this, we heat shock the bacteria and put the recombinant plasmid into the same vial as the bacteria.
After transformation and enzyme production, we have to purify the enzymes to remove them from the cells and isolate them.
We have to determine how well the enzyme degraded the stain. We can determine this by looking at the effect of hydratase on a stain with a pre-treated fabric and on a non-pre-treated fabric.
This year we focused more on the research trying to find a suitable enzyme. In the future we would like to physically conduct the experiment using hydratase on a stain. We also would like to look for more enzymes and find the answer to whether increasing the solubility of oleic acid would impact its effectiveness.